So here's the beginning of part 4 of this hemoglobin myoglobin series. And I want to talk about ways in which the oxygen carrying by hemoglobin is is controlled. So how does hemoglobin know when to change its or alter in some way its binding affinity so that the oxygen can actually be released where it's needed? Okay. Well, we'll start real simple here. Let's just think in the simplest case. Notice we have the T state in equilibrium with the R state. And what drives the equilibrium is O2. So oxygen binds, and what happens when oxygen binds is a proton, just one proton, and actually it's not even one proton, it's 0.6 protons total is given off as a result. Now, I'm going to just say a few things about this, okay? I want you to just think about a few things here. We're going to apply Le Chatelier's principle. First of all, as this T to R transition takes place, in no way does it generate enough protons to change the pH of the environment. A lot of students say that. They claim it on tests and whatever. If you say that, you are wrong. So don't say that. The pH, remember, this is a highly buffered system. Blood is highly buffered. It turns out that the pH range of the blood may be 7.2 to 7.4, something like that. And that's probably a grave exaggeration. I think that would be in the most extreme of cases. So it turns out that oxygen carrying capacity is very much related and governed by pH. Yes, pH. You heard me say it. So we're going to have to be talking acid-base chemistry. So in the transition from T to R state, there is a shift in that conformation that leads to a change in some pKa values of certain residues, and that change releases protons. So I want you to think about this. So if there's a half a dozen or so side chains for which their pKa changes enough so that they become a little more deprotonated, right? Because you're giving up a proton. Then I want you to think, which way did those pKa's have to shift in order to become more deprotonated? Did they shift up or did they shift down? So I want you to think about that. pH less than the pK, greater than the pKa, deprotonated, right? Well, if the pH is stable and pH below the pKa is protonated. I forgot to finish that sentence, sorry. So if the pH is stabilized because of buffering systems present in the blood, pH isn't going to change because of this, but the pH is going to impact this. So what happened to the pKa's of some of and I'll show you the residues in a minute that get affected, but the fact is their pKa's change when they go from the T state to the R state. And if a proton is being released as the transition of T to R happens, then the pKa needs to get lower so that at a fixed pH you're going to get more and more deprotonated as you do that. So anyway, that's just something to think about. So where does this and why is this important? Well, imagine the hemoglobin coming along and it reaches a place in the blood where there is a lower pH. Lower pH, maybe we're on the 7.2 range, for instance. All right? So lower pH means that there's an increase in H3O plus concentration. And that increase in H3O plus concentration will, through Le Chatelier's principle, because a proton is a product here, right? So if we add more protons to the system, then we're going to push it back toward the T state. And the T state doesn't bind oxygen that well compared to the R state. So what happens? Oxygen's released. And the T state is stabilized at low pH. So the binding affinity of hemoglobin gets worse at lower pHs. As the pH decreases, the increases, as pH increases, the H3O plus decreases, right? And so then you'll move things that away to the R state. So at high pH, the R state is stabilized. At low pH, the T state is stabilized. So low pH, hemoglobin doesn't bind oxygen as well as it does at high pH. There's a name for that. It's called the Bohr effect. It's the effect that says low pH, lower pH lowers the binding affinity of oxygen. High pH increases the binding affinity of oxygen. So let's investigate this a little further. Okay. So we've already talked about the interactions between different subunits, and here we have some alpha 2 subunits and alpha 1 subunits, and there's some key interactions that take place in the alpha chains and the beta chains that are part of the source of the change in the pKa's that are involved. Okay, and so there's this lysine that makes a salt bridge with this C-terminal end, and there's over here we have the N-terminal end, and we have another lysine and an arginine that are making some key interactions. Here's another histidine, aspartate. Those are making key interactions and stabilizing the T state, because those are very favorable, these salt bridges. So the T state is stabilized by those interactions. Then, when oxygen binds, these things shift away from each other, and you see a shifting of a lowering of the pKa's for these, because when they're not close to a negative charge, then the positive charge that they could possess is not as stabilized. So the pKa shifts to a lower pKa when it's in the R state. So in the T state, the pKa is a little bit higher, only because it's better for it to stay protonated so it can make these favorable positive negative charged interactions that you're seeing here. So here's one, here's one, here's one. And so when you get the shift then from the T state to the R state, these things shift. Their pKa's change because their local environment changes, and a proton is lost. 0.6 protons is lost. And I mentioned then that by changing the pH, the binding curve changes quite a bit for hemoglobin. So this is an example of a few where you have here's like straight on 7.4 physiological pH, The lower pH here has a worse binding affinity. Less is bound at the same concentration. So this the PO2 of 20 torr is right, I think, is your muscle, like in your muscle, your resting muscle, concentration of oxygen. And so you can see that at a lower pH, there's a lower amount of oxygen that's bound, so it got released. And as you increase to the higher pH, more is bound and held tightly. So where this comes into play is when people are exercising, maybe they're going for a run, their muscle tissue is making use of glucose, and in the process of making use of glucose, the muscle tissue generates lots of carboxylic acids. Okay, and when there is a lack of oxygen at that point, then the glucose is less efficiently metabolized, leading to even more types of carboxylic acid substances. So when that happens, largely your body is not making heavy use of aerobic respiration, but anaerobic respiration, and you get into something called lactic acid fermentation cycles, and that builds up a lot of the carboxylic acids, pyruvate, lactic acid, and a few other ones that get backed up along the way, and it lowers the blood pH in those regions. So when the hemoglobin comes and interacts with those lower pH blood regions where there's lots of metabolism going on, where there's a higher demand for oxygen, then the hemoglobin hits those areas of low pH, and the binding affinity for the oxygen drops because the T state's being stabilized, so it lets go of the oxygen, which then can diffuse across the vein, the artery wall, and be picked up by and stored by myoglobin until it's needed. So that's kind of how it works. When you have low pH, it usually means that you're undergoing lots of metabolism and you need more oxygen. So it's one way that your body talks to hemoglobin. Low pH causes the blood hemoglobin to dump some oxygen as a result. And that's because the T state gets stabilized at the low pH. That's a Le Chatelier's principle thing, kind of tying back to what I just showed you here. As you lower the pH, you push it back this way. So in the lung tissue and closer to the lungs, where there's a higher concentration of charged hemoglobin, you won't see a low pH. It's like in your deep tissues of your leg muscles and stuff like that, where you see this happen. So this is called the Bohr effect. pH influences oxygen binding by hemoglobin. So there's the Bohr effect, and now we can talk about how does CO2 get carried out from your tissue into your lungs. And that is a combination of things. Turns out that some of it can be carried out as carbamates. Whoops. I meant to put that as my pointer, as carbamates. And so CO2 can attach to primary amines, forming something known as a carbamate. And so the hemoglobin is in such high concentration, it can smuggle out lots of the CO2 as carbamates. There's also a good deal of CO2 that gets exported as dissolved bicarbonate. So that is a process catalyzed by an enzyme called carbonic anhydrase. And so some of these things, the formation of the carbamates, the CO2 as it gets dissolved and converted to bicarbonate, releases protons. And these are at a much greater level than just simply the proton that gets transferred when you go from the T to the R state. And so they, along, but to a much lesser extent, these things can help generate lower pH areas. And of course, CO2 is the product of aerobic respiration. And so if there's a lot of respiration going on, there'll be a lot more CO2 in those areas. So this just shows a little cycle. I think it's pretty self-explanatory, so I'm going to let you study it yourself and see if you can kind of trace what's happening. Essentially, just to kind of give you some orientation here, you got the lung where you're breathing in oxygen. Well, I'll just go through it. You breathe in oxygen. The hemoglobin in the T state picks up oxygen, becomes transferred to the R state, moves through the blood, reaches places where maybe the PO2 is, not the PO2, where the pH is lower because you're farther away and you're in respirating muscle. So the pH is lower, so it dumps the oxygen in where the T state is stabilized. Then up in here, you can see that CO2 can cross out of the muscle into the blood. It can be converted to, CO2 can be converted to bicarbonic anhydrase in water to bicarbonate, where it can move freely through the water. Helps buffer, by the way, the blood pH. So it actually serves multiple purposes. CO2 helps buffer the blood pH. But then a carbonic anhydrase, as it gets closer to the lung, can reconvert it back to CO2, which then could be exhaled from the body. And then, as we said before, the protons that are part of this process of this conversion can contribute to the Bohr effect because they're in significant levels. I grabbed this one from the book. This top part came from the book, and that person said this, and I just don't think that's a major contributor compared to the protons that are generated as CO2 is converted to bicarbonate. Essentially what's happening is CO2 is converted to carbonic acid, which then can dump off a proton to become bicarbonate. So that's essentially what's happening. And so the proton can be used to help, again, lots of CO2, lots of bicarbonate formation happening in places where there's lots of exercise happening, lots of respiration, and so the acid form of hemoglobin, which is the T state, can be stabilized then, and so oxygen affinity is lowered, and oxygen can be dumped off where it needs to be dumped off. So what we have here then is another set of binding curves and another opportunity to show other things that can also impact the binding of oxygen by hemoglobin. So there are several different situations, and I just want you to first look at it again, just to remind you, Y is just theta, the fractional saturation. So I want you to look at each of these curves. Okay, we have stripped hemoglobin, so this is out of the blood, stripped of anything associated with it. Here, the stripped hemoglobin, CO2 was added back, so CO2 had an impact on binding affinity. And then 2,3-bisphosphoglycerate, 2,3-bisphosphoglycerate okay, 2,3-bisphosphoglycerate, is a small 3-carbon phosphate containing molecule that actually is a chemical cousin to one of the glycolytic molecules that are in the pathway. It, in fact, gets converted by from 1,3-bisphosphoglycerate. I'll show you where that comes from in a minute. So 2,3-bisphosphoglycerate, if you notice, you add hemoglobin plus 2,3, you change the binding curve again, and then when you add both CO2 and 2,3-bisphosphoglycerate, now you get a binding curve, this black here, that matches kind of what you would see in whole blood. So it suggests that if you look at this, hopefully I want you to rank these in order of binding affinity. Which has, of all the species here, which has the, just we'll say 1 is the best oxygen binding and 5 is the worst. So if we were to rank them, go ahead and do that. See if you can rank them just by looking at the curve. And then I'll rank them and see how yours matches up with mine. You can even turn off your video now and work on your piece of paper for a minute if you need to. I can wait. Okay, so the one with the best binding affinity is stripped. So that's 1. Here's 2, here's 3, 4, and then 5. So just in order. So as you shift in that direction, you get worse binding. All you got to do is just look at one, just pick a concentration of oxygen. You can see this one has nearly 50% bound. And then these drop quite a bit worse if we look at the same concentration, much less bound. So something about whole blood destabilizes the oxygen binding in hemoglobin quite a bit. And so by looking at this, maybe it's the combination of 2,3-bisphosphoglycerate and CO2. We already talked about the role of CO2 and helping lowering the blood pH, and that can impact through the Bohr effect whether or not hemoglobin is in the R state or the T state. And so what about this 2,3-bisphosphoglycerate? What is it, and why does it make sense that it plays a role in this process? So first, let's see what this 2,3-bisphosphoglycerate is about and where it comes into play. So while you were ranking your curves, your binding curves there, I went off into the internet and I found a picture that showed glycolysis, the steps of glycolysis, which is the first part of the breakdown of glucose that gets us eventually to ATP, what we're after. So if we look here, I want you to take a look. 1,3-bisphosphoglycerate. So 1,3-bisphosphoglycerate gets converted into 2,3-bisphosphoglycerate. There's a mutase that can help, or a transferase that can help shift the phosphate. In essence, it doesn't quite do it this way, but it's like it shifts the phosphate from the 1 position to the 2 position. And so when you get a buildup of some of these glycolytic metabolites, that's because pyruvate builds up. So there's lots of needs, so glucose is being used, but pyruvate, in the absence of oxygen, will form lactic acid, and it will forego what is known as the Krebs cycle, or the carboxylic acid cycle. It's got a bunch of different names. TCA cycle, you might know it as. Anyway, I'm tired. my oxygen levels are probably getting really low right now. It's only 12:16 a.m. So I got to finish this before I can go home. I'm still in my office. Anyway, so pyruvate, in the absence of oxygen, it doesn't get utilized very well compared to when oxygen's present. So the pyruvate then builds up, and then that leads all these suckers to build up. And so when this builds up, because of lots of metabolism, because of exercise or whatever, then you get a bigger buildup of the 2,3-bisphosphoglycerate, which is kind of a way to talk to it talks to the hemoglobin and connects it to glucose use and metabolic need. So you get high levels of 1,3-bisphosphoglycerate. Well, you're going to get high levels of 2,3-bisphosphoglycerate, and that will lead to stabilization of the T state. So here's 2,3-bisphosphoglycerate right here, and it's relatively small. It's negatively charged, and it's, as I just showed you, it's intermediate in glycolysis, and it fits in the hole. This is just another sort of picture of the same thing. You can see the 2,3-bisphosphoglycerate binds into the hole of the T state, but not the R state. So when it's bound here, it stabilizes the T state by getting in the way of the clamping down that would have to happen there to switch it over into the R state. So it occupies this little hole and prevents the or impedes the R state as from forming. So the oxygen then can be released when it hits areas of high 2,3-bisphosphoglycerate. It's kind of neat. Think about that. All the different ways that you can control oxygen transport through hemoglobin. If oxygen was just purely dissolved, you would have one concentration. You wouldn't have so much control over it. And by having a protein involved, now you have control. You can tell it where to selectively dump the oxygen, so it's not a one-size-fits-all sort of treatment. It's areas in your body that have low pH, high CO2 levels, have high metabolic need, as evidenced by seeing some glycolytic metabolites building up. All those things are indication that oxygen's needed in a certain area, and so that's a great cue for then push to cause the release of oxygen from hemoglobin. So BPG levels are one way that your body can adjust to different concentrations of oxygen as you're exposed. So if we look here, here's the black is myoglobin. So you can kind of see PO2 levels in your tissues are low. Well, look, myoglobin, you're still at pretty high levels, right? So it turns out that regardless of where your PO2 levels are, myoglobin has a much better binding constant than does hemoglobin, and so you're at the saturating levels pretty much no matter where you're at when in the hemoglobin sort of the myoglobin curve. But if you go ahead and look at what happens then when you live in higher altitudes compared to sea level. So let's look at the difference. Here's the difference in concentration of oxygen at sea level and at 4,500 feet, so at a higher elevation. And if you compare the levels of BPG that are present in individuals at both places, you will find that if you measure and this is after they've acclimated and been there a while, and the new equilibrium of their body is reached, depending on all the stressors that are influencing it, then you have 8 millimolar of BPG at the higher concentration than at the higher, not concentration, elevation than you have at sea level. So that means that with more circulating BPG, that you would be able to have more oxygen delivered throughout your body to compensate for the lower concentration. So it helps that adjustment. So that's one way in which your body adjusts the altitude. I find it kind of interesting. This kind of makes me think of a question that I've asked before, kind of the opposite. And it's a little thought question. I want you to give it some thought too. So I mentioned this before. Oh, maybe I didn't. That may have been on a video, actually, that didn't work out. I made all these videos so many times before I got things to work. Anyway, Michael Jackson. In the 1980s, I recall hearing of a situation where Michael Jackson was, he would go to sleep in a hyperbaric chamber. And the idea was, you have more than just 100% oxygen, but you would have higher pressure, increased pressures of oxygen too, so you can supersaturate things. And maybe part of the deal with the hyperbaric chamber is that you actually could get oxygen to move through your skin and other places too, and kind of infuse your body with oxygen. But when we think about 100% oxygen, boy, what would the PO2 be? I'd have to look that up. I'm not sure. Anyway, think about what might your body do when you're not at a place where there's a decrease. Like here's a decrease. What would your body do under normal conditions where you have an increase in oxygen concentration? Maybe you're out here somewhere. Well, maybe it wouldn't make a big difference, because at some point, even right at sea level, you have just the amount of oxygen available is enough to saturate your hemoglobin binding sites already. Whereas maybe you see a bigger sort of impact on oxygen when you go up where the oxygen levels are much higher, much lower, and you're actually not as near to, like if you look at where you are in the saturation curve, maybe that's what, 0.85 or something like that, where up here you're almost at like 98% or something like that. So if you go in a hyperbaric chamber, you might be closer to 100 or more percent, or not more percent, you can't get more than 100% on binding, but you'll really get super close to 100% at the higher concentrations of oxygen. And so the question is, what might have happened to Michael Jackson's body as you saw the adjustments? Would you see maybe who knows? What do you think you would see in terms of BPG concentrations? What would you think you would see in terms of T state, R state conversions? What would the pH effects be? How would the Bohr effect maybe be impacted by that? I'd like you to give that some thought. Could be a good test question, maybe. Just something to think about, I suppose. So it kind of, while I got this curve up here, I'm going to use it for a completely different reason. I wanted to just take a quick look at, whoopsie. I got to do something here real quick. I got to clear off all this crap. All right. Now I just want to let's just look at we can look in this region right here, okay, where there's not a big difference. So let's just say at sea level, you're out for a run, and you decide that, hey, I really want to try one of those breathable oxygen things. So you pull out your can of recreational breathing oxygen and you take a few hits of that 100% presumably oxygen. Will that have an impact at sea level, for instance, on carrying oxygen to your tissues? Well, a few things I want you to think about. First of all, if you went much higher in concentration from where you're at, if you're at sea level where pretty much there's enough oxygen in the air to pretty much saturate, for the most part, 90 something percent of your hemoglobin, then would you be able to do how much better could you actually do? There's only like a fraction of a percent or two of unbound hemoglobin to even bind to. And so you got to wonder, would taking a hit or two or five or six or whatever make a big difference, assuming you have normal lung capacity and everything's working. I mean, if people have emphysema and stuff like that, they got impaired lungs, and so they're going to have troubles for different reasons, and that's where the oxygen therapy can actually be pretty helpful. So would it make a difference? And I'm guessing at sea level, it very well might not. But what about, if you're already at a place where you're at elevation? Well, people have to use oxygen once they get up, if they're climbing the Himalayan mountains and climbing Mount Everest, for instance. At some point, they have to start taking cans of oxygen with them, and that's really required. So I imagine at some of those higher altitudes, maybe a huff from the old recreational breathing oxygen would potentially, I could see maybe it's possible it can help. For how long and the duration and how effective that would be, I couldn't guess. I'm not even sure what to expect there. And then think about doping, though. Now, doping is a different deal altogether, because you are actually increasing the amount of oxygen carrying capacity that you can have in your blood. So you're limited by solubility. There's only so much oxygen that can be dissolved in your blood. And once you reach that threshold, you can't get more. However, if you could get more red blood cells with more hemoglobin added to your system, then you increased your oxygen carrying capacity. And that's the whole concept behind blood doping. It increases the number of oxygen carriers versus a lot of the other strategies we saw, which was to increase oxygen intake itself. And I'm not so sure those are going to make as big of a difference because of this problem right here. You're already at close to saturation in hemoglobin binding. So adding a higher concentration won't have as much of an impact because you're already using all your sites. But if you could add more binding sites, more carrying capacity, that would make a difference. When we go back to that initial video that I showed you. So I mentioned before that people can study hemoglobin by looking at its spectroscopic UV vids sort of absorbance. And the Fe+2 form without oxygen has a nice absorbance band at 429 nanometers. And when oxygen binds, it alters the environment around that heme, and you see a shift from 429 nanometers to 414. So by looking at that shift and quantitating it, you can actually do these binding assays by looking at the shift as binding happens. And so I went looking on the internet and found someone who actually was nice enough and kind enough to post the different types of hemoglobins that you can find: deoxyhemoglobin, oxyhemoglobin, and methemoglobin. Now, methemoglobin has a different iron in it. It has Fe plus 3. And Fe plus 3 is not very functional. I mentioned that Fe2 plus 2 plus when iron binds, when oxygen binds that iron, you get the shifting into that sort of Fe plus 3 O2 minus sort of transient species that helps hold on to the hemoglobin. And but the methemoglobin is just where Fe plus 3 is just there. You don't have Fe plus 2, and Fe plus 3 won't bind oxygen very well. So methemoglobin, actually, it's got kind of a yucky reddish color, a little different than when you have... Well, if you want to see what methemoglobin looks like, just go to Albertsons. I find that every time I've ever bought meat there, it's always like spoiled or something, and so I don't like going there. But anyway, their meat sometimes, at least the one on Broadway, is quite suspect. And you can definitely see methemoglobin and metmyoglobin present as you look there. It's got a kind of a yucky, not appetizing look to it. But anyway, as oxygen binds, you see the conversion from the deoxy form to the oxy form. There to there. And so you can quantitate. Here's another way to look at it. So you can quantitate the shift as you go from deoxy to oxyhemoglobin, and that's how people can do the binding studies that we just spent a whole bunch of time talking about. All right, and then mutations in hemoglobin can lead to disease, and one of them is sickle cell anemia, and sickle cell anemia is actually the result of a glutamate at position 6 mutation, where it goes from glutamate 6 to valine at that same position. So sometimes I would write this in shorthand notation. I would say glutamate 6V. That's how I would name that mutant, and that's in the beta chain. And so that valine now causes heme to be bound different, and it causes the morphology, the general morphology of the hemoglobin to change, and it actually can form strands, and like little, and so you see these sickle cell red blood cells as a result. People who are homozygous, in other words, it's a recessive trait, and if they have both the recessive alleles, it's fatal. They just can't. They're gonna die essentially from poor oxygen handling, and so in a way they suffocate. Though there is, and you've probably heard about it, there's an interesting little benefit though. When you're heterozygous, you have one allele is producing the mutation with the sickle cell, and the other allele is not. And so what you get is sort of a hybrid. I guess in some ways it's like incomplete dominance. What you get is functioning but not super great functioning red blood cells. And when people are infected with malaria, which essentially targets the red blood cells, the malarial protozoans that cause the malaria, they don't survive very well, and so there's a big resistance to it. So it turns out in some places where there's a high degree of malaria, which can also be lethal, there's a lot of selective pressure then for these heterozygous sickle cell folks because they don't succumb to the malaria. And here's the strand formation that I was mentioning. When you get the mutated form in the beta subunit, you get these weird strands that kind of almost like a fiber formation. And that's not what hemoglobin's supposed to do. But that's what it does in sickle cell anemia. So it's really a single point mutation. That's what we call it. A point mutation leads to a corruption of the protein structure and therefore its function. So there you have it. Hemoglobin. It's what's for supper. That's what I would say if I was advertising, trying to build awareness for hemoglobin. I'd say hemoglobin. It's what's for supper. But other than that, it just carries oxygen. It's pretty cool. And it's a little molecular machine that does pretty amazing things in an amazing way. And we're going to study the tar out of this sucker in class, where we're going to study it using the VMD and some other, maybe WinCoot. It depends on what program I choose to use. But anyway, you'll get a chance to study this structure and get to know it a lot better. Cool.